Okay, welcome back to part 2 of Chapter 8, where we are going to talk about antivirals and some of the beneficial uses of viruses, namely gene therapy and cancer therapy. And so on antivirals, there are obviously a lot of different antiviral drugs out there. I'm not really interested in focusing on names and specific mechanisms of all these different drugs. I want to talk about this more generally in terms of different kind of modes of action, different parts of viral replication cycles that could be inhibited by antivirals. Um, and so, in addition to vaccines, antivirals are a very useful tool in fighting viral infections. And basically, all of the antiviral drugs that we have work by inhibiting one or more stage of the viral replication cycle. And you'll remember we talked about these 7 parts of that cycle: attachment, penetration, uncoding, replication, assembly, maturation, release. Um, and you'll see this cartoon popping up on the next few slides, but it's showing kind of a generic version of a viral replication cycle from attachment to penetration, etc. And you see some examples of some of the specific existing drugs that work at some of these different stages. So I want to start by talking about drugs that would interfere with attachment, penetration, and encoding. And really, if you can stop attachment, that is the optimal way to inhibit a virus, right? Because you're preventing the virus from even gaining access to the cell initially. There's going to be no damage if it can't even get in, no replication at all. So that would be ideal. And there are quite a few drugs out there that inhibit attachment. You see some examples here. In terms of replication now, there are some drugs that specifically work by inhibiting the nucleic acid replication of the virus. And one thing to keep in mind, especially when we talk about replication here, is one challenge with these drugs and with viruses in general is because they are using host cell mechanisms to propagate their replication cycle. We generally don't want to treat with drugs that are just going to generally inhibit important aspects of normal uninfected cells, right? We'd like to try to target these drugs to specifically just affect the virus or just affect the virally infected cells. Um, so in terms of replication, nucleic acid replication, this usually comes down to using what are called nucleoside analogs that are particularly effective for viruses that may have a reverse transcription type step. But nucleosides are basically just DNA or RNA nucleotides without the phosphate group. So remember, a nucleotide is sugar, phosphate, base. So we're missing the phosphate. But these nucleosides can be converted into nucleotides during RNA or DNA synthesis. And so these analogs are chemicals that physically resemble nucleosides. They can actually get converted into nucleotides, but those nucleotides that are generated from the nucleoside analogs are now incapable of forming new phosphodiester bonds. And so if they get incorporated into a growing DNA chain, they basically act as a chain terminator. So they're going to block production of new DNA. And so often this is being carried out by a reverse transcriptase, but really this could be applied, right, for a DNA polymerase would have a similar problem with these nucleoside analogs as well. So sometimes you'll see these referred to as NRTI, nucleoside/nucleotide reverse transcriptase inhibitors. But those are pretty common. And so, yeah, inhibiting genome replication, right, is obviously going to be another effective way of preventing the viral replication cycle. if you're talking about a virus like HIV, for example, that needs to integrate into the host cell chromosome, um, another common technique would be to use integrase inhibitors. So these would block that integration step, um, and if the virus is not able to integrate, form that provirus, um, that is in most cases going to prevent it from uh, being able to complete its overall replication cycle and at least vastly limit the infectivity of the virus. And so the last stage is assembly, maturation, release. There are no drugs developed so far that inhibit assembly, um, but there are commonly protease inhibitors that can be used to limit virus maturation. So we see this with HIV, but it's common with a number of other viruses as well, where initially when the virion is assembled, there'll be these polyproteins that have to be cleaved by proteases to get the mature infectious virion. And so protease inhibitors would block that step from taking place. And then release could be a target if it's a situation where viral proteins are facilitating the release. So if it's just, the virus is just being released by just normal exocytosis, you typically wouldn't want to block that because that's going to affect exocytosis in, normal uninfected cells, which could be very detrimental or life-threatening. But if there's a specific viral protein that could be targeted that's assisting in that release process, as is the case with influenza, there might be some drug possibilities there. All right, so you're probably getting the idea, right? Most of these antivirals tend to be specific just for one or maybe a small handful of viruses, right? They're just because viruses are so different in terms of how they're completing their replication cycles. There is no, magic bullet antiviral that's going to be effective against all different viruses. So aside from the types of antivirals we've been talking about, another more kind of general approach would be, instead of targeting the virus directly or its replication cycle directly, to try to just boost the immune system. Kind of ties back in with our discussion of vaccination. So there has been some success with elevating levels of cytokines like interferons to increase that immune response. You might remember cytokines are those very potent chemical signaling molecules, right, that activate these pathways in the cell that basically block viral translation or protein production and are slowing down viral infections in that way. So that has been shown to work in certain cases, but because these cytokines are so potent, it can be dangerous and it can be easy to overdo it. We talked about the dangers of cytokine storms. I'll bring that back up again here in a minute. So it's really not ideal, but can be useful in certain instances. Okay, so let's finish this chapter out by talking about some ways that viruses can actually be used in modern medicine for other purposes. And the first purpose I want to talk about is what's called gene therapy. And so the whole idea with gene therapy is to try to modify an individual's DNA, replace or correct a mutated defective gene that may be associated with disease. Right? So, we have some very sophisticated techniques now for modifying DNA in the test tube or in the lab, but the challenge is still really, okay, well, how do we do that in a living person's cells, and in particular, how do we do it in the correct cells? So this is where the virus comes in, is we can actually engineer viruses to act as the delivery system for these corrected versions of the genes, right? Because viruses are specialized at accessing cells, and in many cases integrating into the host cell genome. So they're very good, make a very good DNA delivery system. And so let's see, let me just jump ahead here. So there's lots of different viruses that have been used for gene therapy, but one very common one is the adenoviruses, which just cause usually mild respiratory, sometimes gastrointestinal symptoms. These are double-stranded DNA viruses, got a broad tropism, meaning they can infect lots of different cell types. They usually gain entry through endocytosis. They disassemble at the nuclear envelope, and then the genome replicates inside the nucleus. And so the general idea here is you would remove a gene from the adenovirus genome, and typically the one that gets taken out is this gene called E1A, which codes for a transcription factor, viral transcription factor. And you would replace that with the gene, therapeutic gene that you want to correct in the individual. And then you simply introduce the virus to those cells, either cells that have been taken out of the individual in question, or the virus is introduced directly to the body itself and allowed to access cells in vivo. But the whole idea is, right, the virus goes through its replication cycle, and in getting the viral DNA into the nucleus, it is carrying that therapeutic gene along with it. That gene will then be transcribed and translated, and we produce the correct version of the protein for whatever is encoded for by that particular gene. And if we're able to do this in the appropriate cells, we could theoretically cure the problem, right, caused by the mutated gene. So, there are some limitations here. Because you're replacing a gene in the viral genome, and that genome has to be able to fit inside the capsid, you're limited in terms of basically how big the new gene that you're introducing can be. So you have a fairly strict size limit there, so it may not be a good approach for larger genes. In the case of adenovirus, this is not a virus that integrates its genome into the host genome, so it's really just going to be temporary, right? You may get this short-lived beneficial effect, but may need multiple injections or repeated introduction of the viral gene therapy in order to maintain that quote-unquote cure. And another significant challenge, and this could be true for really any viral vector used for gene therapy, is because these are human viruses, they can't elicit an immune response. And so one of the very early famous gene therapy trials was using adenovirus. And a young patient named Jesse Gelsinger famously died because the adenovirus vector that was being used was apparently one that he had probably been infected with before, and the introduction of this high dose for the purpose of gene therapy caused his immune system to overreact, right? Basically induced a cytokine storm, and he ended up dying due to that overactive immune response. So that is always a potential danger with gene therapy as well. But since those early trials, we have come a long way, and there are now thousands of clinical trials underway for various gene therapy applications. So there's still really just a small handful that have been approved for use in humans, but without doubt there's going to be a lot available in the near future. One famous example that's been used just in the last year is gene therapy treatment for sickle cell anemia that seems to be showing great promise in quite a few patients. The second main beneficial use of viruses I want to talk about is in some anti-cancer therapies. And so, if you're not familiar with cancer, of course, these are human cells, right, that are replicating out of control. Very difficult to treat because they are human cells. Traditional therapies would be things like radiation or chemotherapy that will kill all dividing cells. So you end up with some very serious side effects, losing your hair, severe nausea, etc. Um, so the idea with using viruses is we may be able to, again, engineer viruses that would only infect cancer cells, or that might be able to essentially deliver drugs to cancer cells, in essence. Um, or the virus themselves may be able to infect and kill those cancer cells. So there are some pretty spectacular results that have been obtained with these methods. So you'll see this called virotherapy. The viruses used in cancer therapy are often called oncolytic viruses. So viruses that can kill those cancer cells that become infected. And there's all kinds of cool tricks. I won't go into all the details here. I get into it a little bit more in my cancer biology class. But there are some cool engineering tricks that can be used to try to design viruses that will only infect cells that express a specific viral antigen, right? So you would basically introduce the virus into the patient. The virus is looking specifically now for virally infected cells, or cancer cells. Yeah, sorry, I'm saying that completely wrong. Cancer-specific antigens, not viral antigens. So the virus is specifically looking for cancer-specific antigens and can only infect those cancerous cells. So there've been some mixed results, but as I said, there have been some really amazing apparent cures using some of these oncolytic viruses. So just to give you one example, gendocene is this oncolytic viral therapy that was first approved in the early 2000s in China, primarily for head and neck squamous cell carcinomas. But again, this is using an adenovirus vector that had been engineered to contain a gene called p53, which is important for inducing apoptosis. And in this case, they were having to inject the virus directly into the tumors to try to make sure it was getting specifically to those cancer cells. So they were doing an injection once a week for 8 weeks. In addition to the viral therapy, also giving sort of traditional radiation therapy. But this p53 gene often gets mutated in many cancers, especially many head and neck carcinomas. So the idea is the combination of the radiation with the virus is going to increase tumor cell death. And in fact, they saw by adding the virus on top of the radiation, we're getting about twice as much cancer cell death as with the radiation alone. So, in this case, not a magic bullet cure by any means, but, most patients are going to be happy with their tumor being twice as small as it would have been after conventional therapy. So it's at the very least going to buy additional time and, may make other therapies or surgeries more effective if you can shrink that tumor more dramatically. Another kind of variation on this theme is what's called virus-directed prodrug therapy or enzyme prodrug therapy. But the idea here is the virus has been engineered to have genes for certain enzymes, and you would introduce a prodrug into the cancer cells, then allow the virus to infect. And the virus is, once it infects the cancer cells specifically, it will bring the gene for these enzymes along with it. The enzymes will then convert the prodrugs, or the pre-drug, into an active form that will facilitate killing of the cancer cells. The prodrug was probably delivered to all cells, so here you have some normal cells, non-cancerous cells. They've got the prodrug, but because the virus is engineered so that it can only infect cancerous cells, displaying those cancerous antigens, in the normal cells, the prodrug stays a prodrug, whereas in the cancer cells, the enzyme brought by the virus converts the prodrug, in this case, into a nucleoside analog, which is then, as we talked about in previous slides, going to block DNA replication. And obviously that would be an important part of cancer cell division, right, going through the replication cycle. All right, we'll stop there.